Yeah, so the next presenter is Nathan Zammit from Harvard, who's going to talk about regulatory T cells guard against thermogenic dysfunction in brown adipose tissue. Um, if Nathan is there,
I am. Can you see me?
yes, it's perfect. Yeah.
Great. So first of all, it's very exciting to be a part of this worldwide international conference. So good morning, good afternoon, good evening. And I'd also like to take the opportunity to thank the organizing committee for giving me the opportunity to present my work to you all today, which is titled Regulatory T cells in brown adipose tissue safeguard thermogenesis by restraining interferon-producing lymphocytes. So, as you may all be aware, adipose tissue plays an important role in regulating whole body energy homeostasis in the body. And this is mediated by a number of adipocyte cell types, the most familiar of which are white adipocytes, which function to store excess energy in large lipid vacuoles for times of need. And they can be found in multiple compartments of the human body, such as the visceral adipose compartment in the stomach region, or the subcutaneous compartment around your thighs, arms, and legs. But in contrast to this cell type, there are another lesser known brown adipocyte cell type known as brown adipocytes. And instead of storing energy in times of need, what they do is to liberate stored energy as heat in a process known as thermogenesis. And because of this drastically different function of these cells, they look very different under a light microscope. For example, uh, they have multi-ocular lipid droplets and large numbers of mitochondria, which renders, following H&E staining, samples of these tissues to look more reddish brown under the microscope, and hence their name. So brown adipose tissue, as I mentioned, plays an important role in thermoregulation, particularly in neonates. And this is non-shivering thermogenesis, independent of shivering thermogenesis, which is mediated by the muscle. And how this comes about is that following sensing of cold at this level of the skin by the brain, this triggers a sympathetic outflow from the brain and release of norepinephrine to brown adipose tissue depots. The brown adipocytes can sense this via their ADRB3 receptor, and this triggers the upregulation of a number of proteins which are instrumental in producing heat. And one of these key genes is known as UCP1 or thermogenesis. And without going into detail, how this protein generates heat is by short-circuiting the electron transport chain, which is normally utilized to produce energy or ATP. And this release or this short-circuiting of this potential energy gradient produces chemical heat. And because this process of fueling mitochondria to produce ATP or heat energy requires a lot of energy through the breakdown of glucose and free fatty acids in the beta oxidation and citric acid cyclic pathways, it is now recognized that these depots may also play an important anti-diabetic role in adults because these tissues suck up a lot of glucose and free fatty acids from the blood. And indeed, a number of studies in humans have shown that high levels of BAT activity tend to correlate with lower levels with reduced BMIs. And so this really lends to the question whether preventing or increasing BAT function may be a novel strategy to treat obesity and metabolic disease. And so a straightforward question that we had was, because anti-inflammatory regulatory T cells have been shown to play important roles in supporting the function of a variety of organs within the body, such as the skin, colon, and lungs, we asked, do they play a role also in brown adipose tissue, considering that increased BMI is typically associated with increased levels of inflammation. And so to test this question, we utilized a mouse model that allowed us to conditionally deplete Tregs within brown adipose tissue. And following this schematic, you can see that we were able to achieve high levels of depletion within the brown adipose tissue, but also other adipocyte depots. And this led to a marked increase in the infiltration of immune cells, as you can see, marked by CD45 in the brown adipose tissue depots, as well as marked changes in the transcriptional profile of Treg depleted brown adipose tissue compared to non-Treg depleted brown adipose tissue. And specifically, this was characterized by an increase in inflammatory transcripts associated with the interferon response pathways and a downregulation of transcripts involved in metabolic pathways, particularly oxidative phosphorylation. And what I really want to highlight here is that these large changes in gene transcriptomes was largely specific to brown adipose tissue, as if you look at other fat depots, such as the subcutaneous white adipose tissue depot, this magnitude of changes was drastically reduced. So next, we wanted to test whether these transcriptional changes resulted in any functional differences. And to do this, we utilized indirect calorimetry, which allowed us to measure the energy expenditure of mice over time by measuring the oxygen usage of these mice and carbon dioxide production over time. And if you focus on the orange lines, which is mice that deplete for Tregs versus their wild type counterparts in gray, you can see that when we deplete Tregs, this does not alter their energy expenditure at thermoneutrality, nor does it change the energy expenditure when we challenge these mice with 4 degrees. As you can see, mice without T regulatory cells are able to ramp up their energy expenditure. But what we do find is that mice without Tregs in bat are unable to maintain equally high energy expenditure levels equivalent to what we see in the their wild type counterparts. And indeed, this correlated with a reduced core body temperature, as you can see at experimental endpoint when you compare the orange dots in Treg depleted mice compared to the gray dots. And these reduction in energy expenditure levels or core body temperature in response to cold in Treg depleted mice correlated also with an increase in free fatty acids within the blood of these mice, as well as increased lipid droplets in Treg depleted bat compared to their wild type counterparts. And so these data suggested to us that there was some kind of defect in energy utilization in these mice. And so, as I mentioned in the first slide, because mitochondria are one of the organs within brown adipose tissue that utilize a lot of energy to power thermogenesis, we asked whether there could be a defect within this subatomical structure. And to cut a long story short, indeed, that's what we found via electron microscopy. We found that in Treg depleted bat, there were the mitochondria here were abrogated, and this was characterized by a reduced cristae area in these mitochondria compared to what you normally see in Treg replete mice following cold challenge. And this is correlated with a reduction in mitochondrial membrane proteins that normally decorate these cristae, such as electron transport proteins themselves and UCP1. And so we next asked, well, what is driving this phenotype, this abrogated mitochondrial structure when you deplete Tregs? And as I mentioned earlier, our transcriptional responses of Treg depleted bat showed a enrichment for interferon gamma response transcripts. And indeed, we were able to find an enrichment of interferon gamma transcripts by real-time PCR in brown adipose tissue, but not interferon alpha. And so we really narrowed our search to the interferon gamma response pathway. And this uh, focus was supported by the fact that when we looked at the bat infiltrating cells in Treg depleted bat, many of these were expressing interferon gamma, as you can see for T cells and NK cells at much higher levels compared to what you see in Treg replete bat. And lastly, we could show that when we treated Treg depleted mice with an anti-interferon gamma monoclonal antibody, this was able to blunt the interferon gamma transcriptional response in these mice, showing that interferon gamma was indeed playing a role. But was it playing a role functionally? So we did that test by returning to our metabolic clams and measured the energy expenditure of mice, which is a readout of thermogenesis. And indeed, if you focus on the blue lines, these are mice, Treg depleted mice given anti-interferon gamma monoclonal antibody. And the orange lines are mice that have their Tregs depleted but given an isotype control antibody. And the gray lines are the wild type controls that have Tregs within their brown adipose tissue depots. And what you can see, if you compare the blue to the orange, is a rescue of the energy expenditure levels in antibody-treated mice compared to their isotype controls. And this was also bared out in their core body temperatures measured by rectal probe, as well as a rescue of this lipid utilization effects that we saw earlier by H&E analysis. And lastly, we could also show that this also rescued the structure of mitochondria with a rescue in the frequency of crystal area per mitochondria in monoclonal antibody-treated mice compared to those given an isotype control antibody. So together, these data show that anti-interferon gamma rescues Treg depletion effects in bat. But our next question was, does interferon gamma directly impair the function of brown adipocytes, or is it the other cell types, such as macrophages, that are activated that are then imparting these defects? So to test this question, we simply differentiated brown adipocytes in vitro, in culture, and then added interferon gamma directly to these cells to see if they could affect their oxygen consumption rate by seahorse analysis. And just to cut a long story short, that's indeed what we found. If you look at the red line, which are brown adipocytes treated with interferon gamma versus the blue line of brown adipocytes treated with vehicle control, you can see that interferon gamma blunts their maximum oxygen consumption rate, and that these cells, when we conducted bulk RNA sequencing analysis, had reduced transcripts for the oxidative phosphorylation, which mirrors the findings of our in vivo experiments. And so we next wanted to ask or have a closer look at the Treg heterogeneity within bat to try to get some insight on how depleting Tregs was leading to these large effects in function of thermogenesis in these mice. And so to do this, we isolated Tregs from a variety of adipose tissue depots and subjected them to single-cell RNA sequencing. And essentially what we found was 4 unique clusters of Tregs defined by 4 unique transcriptional profiles, which we labeled as reparative ST2 positive Treg subtype, or a TBET-TH1 type of Treg, an IL-18 receptor 1 positive Treg group, and a quiescent group. And the take-home message here is that we found that these clusters were differentially enriched across fat depots. So if we focus on brown adipocytes, brown adipose tissue in brown, these tissue depots had more IL-18 receptor 1 positive Tregs and less reparative ST2 type Tregs compared to white adipose depots, such as the VAT, which is highlighted in red. And so we next asked, do these Treg subtypes change with cold challenge? And indeed, we did find that there were some changes. So when we subjected mice to cold for 24 hours and looked at the Treg numbers within the different tissue depots, BAT, SAT, or VAT, we found that the IL-18 receptor 1 Treg subtype was preferentially enriched in primarily BAT, but also a little bit in SAT, but not changed in VAT, and the other Treg subtypes were not changed. And indeed, we found that the ligand for IL-18 receptor 1 was specifically acutely upregulated in brown adipose tissue following cold challenge. And so this made sense. And so we next asked, do these IL-18 receptor 1 positive Tregs play a role in BAT function? And so to test this, we utilized the mouse model where we could specifically delete this receptor within T regulatory cells. And this did not delete these cells, but merely just remove this receptor from Treg cells. As you can see that the numbers of total Tregs within this tissue depot at room temperature and at 4 degrees cold challenge does not change. But what it does do is disables the ability of a mouse to maintain their core body temperature following a cold challenge as high as compared to mice that do have IL-18 receptor 1 on their Tregs, as you can see when you compare the solid dots to the open dots here, suggesting a role for IL-18 receptor 1 on Tregs in maintaining mitochondrial fitness in brown adipose tissue. And so lastly, these defects correlated with an increase in interferon gamma transcripts within brown adipose tissue, as well as an increase in infiltrating interferon gamma positive CD4 T cells. And so to summarize, we found that IL-18 receptor 1 Treg cells safeguard the thermogenic potential of BAT by reining in local interferon gamma responses. And essentially, this occurs through the IL-18 alarm and axis, whereby cold induces an increase in IL-18, and this allows the Treg heterogenic population to respond accordingly, increase a Treg population that can appropriately control the interferon gamma responses of infiltrating or local T cells. And when you remove this homeostatic response pathway, this causes an overabundance of interferon gamma and leads to a dysregulation of the mitochondria within brown adipose tissue and reduced thermogenic potential. And so with that, I'd like to thank you all for listening today, as well as the lab where I did this work. Thank you very much.
Thank you so much, Nathan. That was a really fantastic and great story. We have actually a couple of questions regarding this. So Eleanor asked if you were surprised not seeing interferon alpha in instead of interferon gamma.
That's a very good question. I guess I would say I was surprised and I wasn't surprised at the same time. With I don't know if you, with GSEA analysis, a lot of transcripts are shared between the interferon gamma and interferon alpha response pathways, and so it's also always a good strategy to try to deconvolute those. But I guess maybe Eleanor's coming at the question that mitochondria can trigger the cGAS sting pathway through interferon alpha as well.
Absolutely.
Yeah, that's another strategy that uh, where that was a focus of our initial studies as well, and we could show that there was some beneficial effects of targeting that pathway, but it wasn't as pronounced as targeting interferon gamma.
Thank you. There's also a question from Payam asking, like, what is your thoughts about the evolutionary mechanism behind this phenomenon? I would ask also if you see an increase of T-Rex, for example, in animals that are hibernate, for example, are or more exposed to cold. Do you see like enrichment of T-Rex in those animals?
Yeah. And that's a very thought-provoking questions. I guess a theme coming out of our studies within the Mathis lab was the fact that we're seeing these increase in Treg populations occur in response to specifically in tissues that have a lot of mitochondria, such as muscle, brown adipose tissue, and things like that. And there seems to be a conserved role for T regulatory cells to um, control the homeostatic balance in these metabolic intensive tissues. Whether it's responding to cold or responding to interferon gamma signals from stress or other types of stress, such as load stress in muscles. But I think there's a unifying theme there that we're looking into and might be able to be taken advantage of for treating metabolic disease. Um, what was your question? Sorry, Euglene?
Yeah, if for example in animals that hibernate that are like more exposed to cold, you have a like enrichment of this T-Rex or the in those tissues in
Yeah,
comparison with
that's
the
a great
animal.
question. Something that I haven't pursued, but I think would be worthwhile pursuing. Yeah.
Yeah,
Sorry,
there's
answer.
yeah also a question regarding if you have you done trajectory analysis with your RNA-seq data to see if the ST2 population come from the IL-18 receptor or vice versa.
Yeah. Yeah. So we have done some trajectory analysis and it appears that the they split off. So there is some sharing, but if there was a trajectory, it'd be from IL-18 to ST2, not the other way around. But the some cells definitely do come from the quiescent path directly to the IL-18 or to the IL or to the ST2. But there's also like a sub intermediate population which the IL-18 group might also represent.
I will then just do one last question. Thinking about GLP therapy. Do you know how does the therapy affects the population of T-Rex within the tissues? Like meaning, I'm thinking that in the bad you have this transcriptomic profile, but then you give the drug and then you lose this fat. What happened to those T-Rex?
Yeah. That's a great question. So there are some papers showing that insulin signaling on Tregs themselves can play an important role in how Tregs respond to thermogenic challenges. So I'm sure there might be some perturbations there. But if these inhibitors also modify the inflammatory phenotype and the stromal cell compartments of these cells, that they also probably reduce or change the heterogeneity subtypes of these Tregs. For example, the stromal cells within VAT um, talk to the Treg compartments through IL-33 signaling, and definitely whether GLP-1 modulates the stromal compartment and then therefore has unknown effects on Tregs would be an interesting avenue for study for sure.
Thank you so much, Nathan. It was a pleasure to listen to your work.